Good morning, everyone. I'm Simran Costa, and on behalf of Altum Technologies, I'd like to welcome you all to our new Women's Day special virtual webinar series. Our session for today is entitled The Success Saga of Multi-Omics Technologies for Addressing Global Food and Nutritional Security. So, before we begin, I'd just like to give a very brief introduction to today's topic. So, food and nutritional security are an essential goal for the entire global population during climate change scenarios, and these twin challenges can be addressed by expanding the global cropping system by promoting grain and legume crops in India. And recent advances in next generation sequencing or NGS, together with other omics or multi-omics technologies, have significantly reinforced crop research. It became a very powerful tool to produce genomic data, and various other integrated techniques have considerably expanded and deepened our understanding of living organisms, molecular systems. So, multi-omics technologies bring novel tools and concepts that can enhance the precision and efficiency of plant breeding, such as the development of cost effective, high throughput genotyping technologies and its various applications in sustainable agriculture. And these technologies enable plant breeders to genotype many samples in a short span. It is used to implement genomic selection as a routine practice in breeding programs for fast track development of superior crop breeds that are stress resistant while still having a very high nutritional value, leading to growth in the farmer's income. In conventional plant breeding approaches, it takes much longer time to create new improved varieties because this usually relies on phenotypic selection and breeder experience. Moreover, you have complex quantitative traits that have low heritability and are therefore difficult to select. And modern breeding procedures are advantageous because they are more reliable and efficient. So the use of single nucleotide polymorphisms or SNPs in genotyping assays is promising for dissecting genetic variability in cultivated and wild germplasm to identify important abiotic and biotic stress tolerance genes. As genetic markers, SNPs are the most abundant DNA sequence variation which are stable and it can be automated. It's very efficient and it's increasingly cost effective. Therefore, it's more feasible and sustainable to use cutting edge technology to power agronomic development. So, let's understand more about this topic from our speaker of the day. It gives me immense pleasure to introduce Dr. Nisha Singh. Dr. Nisha Singh is an assistant professor in bioinformatics at Gujarat Biotechnology University. She holds a PhD in biotechnology and bioinformatics from NIPB, New Delhi, and postdoc from Cornell University, New York, USA. Dr. Singh also served as DST Inspire faculty at NIPB, New Delhi. Her research interest lies in genomics, proteomics, and bioinformatics, developing cost effective, high throughput genotyping technologies for genetics and molecular breeding applications in diverse crop species, including rice, wheat, pigeon pea, and mango. These technologies will enable breeders to genotype high throughput samples in a short span of time, to implement genomic selection as a routine practice in breeding programs for fast track development of superior crop varieties. Dr. Singh has developed the first patented genic 50K SNP genotyping chip, OSSNPNKS, a single copy gene based SNP genotyping chip for genetics and molecular breeding applications in rice. She worked in several international genome sequencing projects, including wild rice, wheat, mango, jute, pigeon pea, and tea. Dr. Singh has developed the first haplotype map of tea to facilitate the understanding of tea genome evolution and tea metabolite pathways, thus offering an effective germplasm utilization for breeding and tea varieties. She has published 76 international publications in reputed journals. Her H index is 23, and she has more than 2,200 citations and has three patents and two edited books. For her scientific contribution, she has gained several prestigious awards and honors. So, without taking much time, I'd like to hand over the platform to our expert. So, welcome, ma'am, and I invite you to take over the session.
Thank you, Simran, for this platform and opportunity and this wonderful introduction. And this is actually a very good initiative. I must congratulate you and your team for this Women's Day special webinar. And one more thing is that on 11th of February, we have International Days of Women and Girl in STEM. So this is actually, I can say that it's a double benefit, like I'm speaking before 7th March and before 11th February. So first I would like to thank you all and congratulate for this very good initiative. So I will start with my topic, and as Simran has already mentioned, that the topic is on the success saga of multi-omics technology for addressing global food and nutritional security. So I will just take to you a tour of like what are the different technological platform advancement has been made in the last few decades and how it has helped us in addressing the global and food, yeah, and nutritional security, because this is one of the major mandate of global, and this lies under the sustainable development goal second. So in the introduction I would like to emphasize that as we all know that our global population is increasing very rapidly, and it is expected that by 2050 we will be touching around 10 billions population. So to meet this projected demand of growing population, we need a sustainable agriculture system so that we can able to provide the food. But there is several constraint in that because of our climate change, because of our this limited resources in terms of fertile land, also the genetic composition of our elite cultivated variety, and also the with the grain yield. Along with that, we need to have a good nutritional food, because we are eating something but not getting the adequate amount of nutrition, that is termed as an hidden hunger. Okay, so one third of the global population is actually facing this serious malnourishment problem, which is leading towards several kinds of diseases and deficiency in the coming population. So, keeping this view in mind, so the Sustainable Development Goal 2 is saying that we have to have and the hunger. So the first and foremost goal is to zero hunger, achieve food security, and improve the nutrition. We in the green revolution era, we are just talking about the yield, not the nutritional aspect. We are just having to have a high grain yield, so we are more focusing toward the yield component. Now along with the yield component, we wanted to enhance the nutritional attribute in the crops, so that is the yield plus nutrition we have to require for the sustainable agriculture system. So this is one area in which we are working since almost a decade. So I will just take you some of the research which we have done and how we have incorporated this multi-omics technology for the enhancement of better crop production. So the multi-omics technology, if I say how it is, the recent terms have coined, because earlier we are constructed ourselves on a single omics. For instance, it's a genomics, either transcriptomics, proteomics, metabolomics, or ionomics. Now the era of to integrate all these omics and to produce a good crop yields, which is having a high nutritional value, right? So in this case, what is the multi-omics? So om means the universe, the Brahman. So the multi-omics is the cosmic world of different omics, such as genomics, transcriptomics, proteomics, metabolomics, ionomics, and now phenomics. Phenomics is the phenotype of a crop. So if you see this figure in which we are saying that the genomics, which comprises the genetic material encoded in the DNA nucleotide. So with this, we can fetch the information along with the phenotype, and we can conduct the genome-wide association studies. We can study the pen genome study and identify the structural variants present in the different genotypes of this variety. So through that we can able to understand, we can understand the presence absence variants through the genomics. If it leads to the transcriptomic level, that is the expression in which we can identify the differential express gene and network prediction, and also we can perform the single cell RNA. Also we can do in genomics the single cell genomics. And now move forward towards the proteomics. So in proteomics we are doing the protein search and protein expression analysis, structure and functional prediction of that particular protein. We can conduct the analysis of protein-protein interaction, identify the hub protein, also the key protein involved in the interactions, and post-translation modification we can perform. Now come to the metabolomics, in which we can do the metabolite profiling of different tissue samples of plant, along with the comparative analysis of disease versus healthy plant, also stress versus control plant. Okay, so these kinds of analysis we can perform, and integration of all these analysis is collectively known as the multi-omics approaches. So why it is essential? It is essential for the understanding of plant biology with a multiple layer of information, because we are not relying only on the one information. We want to have a multi-layer of information. And why it is essential? Because through that we can accelerate the crop improvement, and it further accelerate, facilitate the marker assisted breeding, and also for the genome editing and genetic engineering of the for the crop improvement and the breeding program. So this is the overall objective and goals of the multi-omics analysis. So the multi-omics is having the two approaches. First is top-down approach, and the second one is bottom-down approach. So the top-down approach is comprises from phenotype to genotype, means we assess the phenotype, and based on the phenotyping we perform the genotyping experiment, means we wanted to decode the genetic information lies in that. And in the bottom down approaches, we first dissect the genetic component, genetic element present in that, differential genetic element, and then we will try to correlate it with the phenotype information. And that is the concept in the breeding program. So basically in the crop breeding program, we are having two types of approaches. First is the top down approach, and the second one is the bottom down approach. So if we talk about starting from the genomics to inomics, I'll take you. So the first advancement happens when we learn how to sequence the genome and decode the information and how many genes are lies in that. And through that, we can able to identify the genes of interest and make a crop improve traits of that particular crop. So for instances, in case of this plant, we have first Arabidopsis thaliana, who has been sequenced in year 2000, and it's a fairly small genome, and it has total 25,000 genes. And after half a decade almost, we have gold standardized genome, in which India has participated for the genome sequencing project, and it has total 36,000 genes and 389 MB of genome size. And then after again 5 years, we have exclusively indigenous pigeon-pea genome sequencing. First legume genome, which has been sequenced in India from NIPB New Delhi and ICRISAT, both have contributed, and the paper has been published in this on the same year. So it is the crop which has been sequenced in India, and its genome size is 852 MB. Total 47,000 protein coding genes are present. But if we talk about now era, now sequencing is very easy task. You can do quickly, but the major problem with that, how to interpret this data. And now we are having hundreds of projects going on in which we are doing the sequencing. For instance, is the Earth Biogenome Sequencing Project. So we have more than 1,000 plants genome which has already been sequenced, and they have the gold standard set of the reference genome. In the Indian context, if I talk about, so we have also involved in sequencing of mango genome, jute genome, and tea genome. So these are the some indigenous crop which India has exclusively do the sequencing alone. So if we talk about the evolution of the sequencing platform, so it started from the first generation, as we all are aware, from the Sanger, Maxim, Gilbert sequencing, followed by the second generation sequencing, and now we are having third to fourth generation sequencing. The basic difference between these sequencing platform is that the yield of the data, also their chemistry, and their library preparation. So these are the major differences in the every generation of the sequencing platform. So now in the modern era, the third and fourth generation of sequencing, you can see we have this very hi-fi system. For instance, is NovaSeq, Nanopore, Promethea, and for the long read sequencing, packed by a SQL 1, 2, and Review platform for the long read sequencing, in which we can have a scaffold level, chromosome level of sequencing platform. So they are actually very good platform. So we will have to have a gapless genome sequence, and the yield and the quality of data is pretty good. So the challenge, if I talk about in the sequencing, is we can have a sequence, but the problem is that because of the high data, we will have a lots and lots of storage required. So that's how the another branch of science emerges, is known as computomics. So in computomics, we have a higher cost with the big data science, so we have to do the analysis on the cloud system, or we need a very good server system to analyze this high throughput data. So that is the major consideration, and also the expertise required to read those information and extract the meaningful information out of it. So if we talk about the assembly strategy, so for the long read and short read, the assembly strategy is entirely different, and also the softwares are completely different. If I talk about the long read sequencing, so it is based on the overlap layout consensus sequence, and for that you can see that the software is Kano, Falcon, Flies, and Flies NG. But for the short read sequencing, it's based on the Kamer method. So you can see it is useful for the spade bias for the sequencing. So these are the two different sequencing platforms. So what is the complete step in the doing freezing the genome? So it's kind of a jigsaw puzzle. For first, we have to have the raw sequence data. We have to perform the quality analysis. If the data is of good quality, we'll go ahead with the assembling of this thing. So initial data, raw sequence in the small stretch of reads, and then after these reads, we have to perform the containing of the sequencing, the assembly into the long pieces of the sequence, and that process is known as containing, and that containing makes the scaffold. And the scaffold, if there is any gap left over, so for how we will fill those gaps, we will perform the polishing of these gaps. So the polishing can be done through the Racon and Pylon. So these are the two most popular software for the polishing, so that we can assign the nucleotide bases in the through AI-based approaches. We can assign these ends. We can fill the ends with the nucleotide, and then finally we have chromosome level of assembly, that is known as pseudomolecule. So this is a complete overall step of the genome assembly. Then how you will see the quality of assembly is really good or bad, how we can assess that. So for that, we have different tools and software. For instances, for the Quast, also icarus is there, so it will tell you the how good your assembly is, whether it is good or bad. So accordingly you can decide and proceed further for the downstream analysis. So this is actually complete goals of genomics. So first you have to sequence, select the plants which you want to do the sequences, whether you want to do the reference based sequencing, or you want to do the de novo sequencing. And then after that you can choose the platform through which you want to perform the sequencing. After that, if you have done the long read, of course nowadays we are doing the long read sequencing. So the long read sequencing, we have to perform the assembly with the respective softwares, and then perform the annotation. Annotation is that, for instances, in case of any plant say pigeon pea is having 47,000 genes. So those 47,000 genes, what they are encoded for, what are those functions they are doing, so that is known as annotation. So we are assigning the functional function of that particular genes, so that is the annotation. And now we have a huge set of data. So the next task is to where you can categorize those data. So the next task is to develop a database of that particular information, because you cannot keep these information in your own system. So you have to have a database so that everybody can access those information, and they can use for further downstream analysis. For example, gene identification, variant detection, SSR, ESTR, indels and SNPs identification, copy number variation, structural variation we can perform. And finally, out of that, mine the information of particular traits which we want to target, or the genes, then further go for the gene editing or marker assisted selection in a breeding program. So this is the overall concept of the genomics and how it's helping the crop improvement. And through this approach we can develop a new improved variety, which is not we are able to do through the only phenomics. So that's how the genomics play very crucial role in the production of superior breeds in the breeding program, and we can have a very good variety, elite variety through this approaches. So now in this line we have already going on several bio earth genome project in which they wanted to encode the sequence of life, and the three major objective they have keep in mind for designing this particular project. The first one is revolutionize our understanding of the biology and evolution through sequencing. We can estimate the conserve, protect, and restore the biodiversity, and also we can create a new benefits, means new variety for the societal and human welfare. So these are the three major objectives for this Earth Biogenome Sequence project. And in this line, in the UK group, they are also doing the Darwin Tree of Life, in which they wanted to read all the information and wanted to construct a tree of life again based on the whole genome sequencing approaches. That's to a gapless assembly, because now we have a privilege to do the gapless sequencing, because earlier technologies is having gap. You can see an example of that. The human genome has freezed gapless in year 2022 and 2023, but earlier we have the human genome sequence data, but they are also having some of the gaps existing on those data. Now we are having the gapless human genome. So similar sort of analysis we are trying to do in other crop species as well. So in the genomics, we have deals with one reference-based or pen genome sort of thing. But now, if we want to do the multiple sequencing of same species, that is known as the pen genome. So what is the application of the pen genome? The pen genome is actually the combination of genetic information of multiple genomes to create actually a composite reference that capture the genetic diversity of that particular species. We can estimate the structural variants within a species. Also, mining those variants, single nucleotide polymorphism, we can perform the association analysis and map those traits. So for that, you can see that in case of plants, we have plant pen genome databases, and it actually started in year 2005 when the Arabidopsis genome has been freezed, and different sequencing technology have been employed for completing this task. So it's having NGS technology, hybrids technology, and other technology platform. And now you can see that starting from the edible crop to the horticulture crop, we are doing this pen genome sequencing, and number of publications has already been published on this particular study. So this is the one study which we are currently doing in collaboration with the ICAR and IPB New Delhi, along with the IHR Bangalore and other partners of ICAR, in which we are just trying to freeze the genome of mango through triose-binning assembly approaches. So actually, this particular variety, Amrapali, is the nothing just a cross between Neelam and the Sherry. So you can see that using PacBio sequel 2, we have this 300x coverage, and we perform the assembly of both maternal and paternal. So out of these two, the Sherry is actually a maternal of this mango, and Neelam is the paternal. And we identified the genes, that total number of genes present in Neelam is 36,143, and in the Shari we have 36,273 genes. So total 900, approximately 900 genes are gained in the Shari, and which is lost in Neelam. So you can see that at the sequence level we can estimate the gain and loss of genes. So these are the two parent, but their F1, the Amrapali variety of mango, is having more number of total genes as compared to the Neelam and the Shari. So the total number of genes is 45,890. So almost 9,000 gain of genes in the Amrapali. So make is more heterotic. So it shows that the F1 is a hybrid, and it has gain of gene, not the loss of gene. So it shows its heterotic superiority. So through this sequencing platform, we can able to perform this type of study, and which is very interesting. And this type of study, trios spinning assembly, we can generally perform for the human genomics as well as the livestock genomics. But in case of plant, if you know the maternal and paternal plant donors, so we can perform this sort of analysis in plant also. And so this is the one study we have done in case of mango. And also using this sequencing information, we have tried to do the phylogenomic analysis and identify the evolution and the synteny between the species, total number of genes unique to species, and total number of genes which is shared between the two species. So you can see that this is a two Venn diagram, and you can see the total number of genes is 39,150 in the mango, and it between the species, it is showing that with the green color, and the unique gene with the red color. So that's how you can perform the phylogenomics and synteny analysis, gain of gene and unique genes present in that. So unique genes make that particular crop unique in that aspect, because these are the genes which is actually contributing to make them unique. Similarly, in case of tea, that was the story of mango, and then we perform indigenous this tea haplotype map, and we perform this haplotype map very first time from the India, and this study is actually based on total 369 diverse set of T genotype, and T has total 4 GB genome size, so its genome is quite complex. And this is the pipeline we have developed in house, and we require lots of high-end server cloud computing. We perform this analysis, and the T has total 15 chromosomes. So this we have developed through coding our inbuilt script. And you can see this analysis is useful for the tree breeding program. And this is the first report from the haplotype map of from the India at the largest scale, because before that, in case of human hap map, has been generated from the UK group, also in the US, and also in the crop, the first haplotype map has been developed by the Buckler groups in the maize. So this is the first report from India side. So we have developed this haplotype map with the used data sets, and also develop a database for the T hap map in which all the information is available. So those who are working on T, yeah, they can get these information easily. It's openly accessed. Now come to utilizing this sequencing information, we can generate the arrays of different platforms. So we are actually stratify arrays into platforms. One is low multiplexing with high sample throughput, the other one is high multiplexing with low sample throughput, and both the platforms have varied applications. For example, low multiplexing with high sample throughput useful for the population structure analysis, foreground selection, diagnostic purpose, and seed quality control. And high multiplexing with low sample throughput useful for the evolutionary study, association mapping, background selection, genomic selection, and diversity analysis. So utilizing these genomic information, we have fabricated the first high throughput SNP genotyping platform. This is a patented technology from India, and we got a lot of appreciation for this work and from the government of India as well. So in this line, we have developed for this genotyping platform for other species as well. In PGNP and mango, we have developed 62K chip and 80K chip for that. So through this chip, it is actually help in accelerating the breeding program. as during introduction Sheetal was mentioning that the conventional plant breeding method based on the phenotypic selection, and it's all depend upon the breeder's experience to develop a new and improved variety. But because of this genotyping platform, we can able to perform the analysis on the molecular level, and it drastically reduce your breeding time. So on an average, one variety with the help of conventional method took so almost 10 to 12 years, but because of this molecular breeding or marker assisted selection in a breeding program utilizing this high density array, we can able to develop any improved variety of interested traits within 3 to 5 years. So that we have done on in utilizing this 52K and 62K chip. We have the other breeders, rice breeders as well as pigeon pea breeder, they could able to generate a varieties number of varieties has already been generated through utilizing through this SNP genotyping platform. So this is the high throughput genotyping platform. And what are these applications other than the breeding program? We can harness the genetic diversity within the species and across the species. So this is one of the very wonderful example. And we can study the population structure analysis utilizing this information, and we have published this paper. And in that you can identify what are the pure seed line and which are having the impurity of the seeds. Admixture population and pure lines we can easily trace through this molecular marker. Also we can study the history of crop origin and domestication from and their ancestor from the wild type, so we can establish the relationship between the cultivated versus wild type. And this is the new thing which we can perform, not only GWAS, we can also perform the TWAS, that is transcriptome wide association analysis. So GWAS is genome proteome wide association analysis, and now recently genome phenome and environment wide association analysis we can perform utilizing this genomic information for the phenotyping. So what is genome-wide association analysis? And these are the steps involved in the genome-wide association analysis. So first we have to collect the diverse set of germplasm which we want to perform the association and identify the trait of interest. So this is the ongoing project from DST. We got the sanction of that. So through the genotyping, we use this 62K CCSNP and NKS chip of Arhar pigeon pea, and then perform the phenotyping of these many traits, which comprises total seed weight, protein content, amino acid estimation, resistant starch, and then perform the GWAS using statistical model. And these statistical model could help us to identify the causative SNPs or responsible for that particular trait. So it's actually a correlation between these, and through that we can identify the causal gene because the SNP is associated with that gene. And then we can understand the molecular mechanism, and further functional analysis through RT-PCR we can validate, and we can perform this. So this is all the systematic representation of GWAS analysis. And utilizing this, we can identify these two OSXDH and SSXT family of protein present in the deep water rice, and through that we can able to develop two varieties of Nigeri Bao, Dhan, and Sargafala for the Assam region. So this is the product through this uh, genotyping platform. Also, we can utilize this for the development of high density linkage map of different traits of the for example the pod number, pod length, and branch pattern. So ultimately it will leads to increase our yield, but we also wanted to look at their nutritional quality aspect like total protein content, micronutrition profiling. So all these kind of analysis we can perform. The thing is that we have the genetic information available with us. We have to perform the phenotyping and perform the GWAS to identify the marker trait association. We can perform the gene and genome based haplotype analysis to establish the relationship between how the domestication happened, and due to domestication events what are the genes we have left behind for that. So we are actually claiming that we have the genetic diversity, so that thing also we can trace through the genomic interventions. Now come to the high throughput phenotyping. We have learned a lot about the genotyping thing. Now the term is how high throughput phenotyping, because to study the correlation between these two, or for example the marker trait association, we must have the genotype information along with the high throughput phenotyping of repeated at least 3 years of data. So high throughput phenotyping is discover the genetic traits that are desired to express, and these actually influenced by the genotype, environment, and the management. Because, for instances, if some particular variety is working well in the Gujarat, but it's not really working well in the southern part of the India, because the reason is the environmental impact. The second thing is management practice. The fertilizers and the extra nutrient which we are adding here in this soil might missing in that soil also may enrich in other components. So that is the interaction which we have to take care. So that's why the high throughput phenotyping multi-location is really very essential to achieve precision breeding. So high throughput phenotyping is now not no more on the manual basis. It is actually AI driven. We have robots and drones which are picking up the data, and then we can analyze those through the a deep learning method. So these are the robots and drones which capture the image in the field, the maize field, and similar sort of things we are doing for the pesticides management and soil analysis and weather forecast. So now the agriculture, if you can say that the agriculture technologies which we try to integrate to identify the key traits which is responsible for that. So these are the various types of high throughput phenotyping platforms available. So what are the advantages of these? So they actually facilitate rapid. They are non-destructive because it's some image based. So they rovers are actually capturing the images only. So we are not harming the plants. It's high throughput detection, addressing the shortcoming of traditional method, because traditional method is based on the manual scoring, and it's a man-made, so errors, the chances of error is quite high, and also it's vary from batch to batch and sometimes person to person as well. We cannot do the traditional method with the real-time monitoring, but because of this AI thing, we can perform the real-time monitoring. It's a large-scale plant traits measure, and we can able to discriminate the cultivars. For example, in a field, if some cultivars is of odd type, so that also we can trace through the AI based, ML based approaches. We can predict the crop yield, selecting the genotype for the trait of interest. So these are the major advantages of having HTP platforms. Now come to the transcriptomics aspect. So I'll not go more detail into that because it's same as the genomics. So these are the transcriptomics approaches, in which, like genomics, we can do the two types of transcriptome. The first one is the reference based, the other one is de novo based, as we are doing the this in sequencing. The major difference is that we are able to identify the circadian pattern within the transcriptome data, also tissue specific expression we can identify, and also we can do the differential expression of gene analysis that is not present in the your genomics things. Now come to the transcriptomics. So in the cell transcriptomics, we can able to capture the single tissue and analyzing those data for the analysis. But still, in this case, the tools are not really very well explored. We have lots of transcriptome data, but the single cell transcriptome data, but analyzing the software, there is only one software is available, is Sirot, but people are working toward development for this area as well. And now come to the proteomics and metabolomics in plant. So basically, in both the proteomics and metabolomics, we are doing two types of study. One is targeted, the other one is untargeted. So the targeted is actually you have certain compounds against that we can profile your metabolites, and the untargeted, it's a kind of, you can say that the de novo, you are doing first time and you don't know what are the all metabolites you are looking for, so you are cataloging all the metabolites present in that, and then you are identifying the biology analysis. So this is the overall concept of the proteomics and metabolomics, and then we can do this correlation analysis with the genomics, transcriptomics, proteomics, metabolomics data thing. So towards the plant proteome atlas, we have databases which is available for the plant proteome databases, in which all the plant proteomes are there. We have cataloged till date, and we can also perform this comparative proteome analysis in certain conditions. For example, in certain salinity stress condition as well as control condition, and identify the different proteins available expressed in under as well as these thing. Also utilizing those protein, we can perform the docking study with our metabolites as well as also with the ligand based study. So these are the things we can do now. Now in this era, we all know that this is the era of AI/ML. So you can predict your protein structure with the AlphaFold, and they last year they got the Nobel Prize for the AlphaFold prediction. And they said that this is the very first time that the computational biology has been acknowledged by the Nobel Committee to for this research. So we can see that at a time we can analyze lots and lots of protein structure. In the comparative metabolomics, also we can perform the LC and GCMS. My student, she is working on the so she is identifying the differentially metabolites present in that. So you can see that in the GC data we have total 65 different metabolites, which is commonly present in healthy and disease one, but they have the unique metabolites in healthy and disease one. Similarly, with the LCMS data, we have identified total 259 commonly expressed in healthy and disease, and uniquely expressed in healthy plant 762, that is 37.3%, and 50% in the disease plant. So you can see the expression level at the metabolite profiling also, and the disease and healthy plants is entirely different and their expression. So we identify some of the unique metabolite which actually contributing towards the resistance, and some contributing towards the susceptibility of that particular disease. We have also ventured into the database and software development for the omics analysis, since we are dealing with the large sets of database data production, because we are working on the multi-omics approaches. So where we can capture all those information, either there is a two-way, we can submit the data, or we can develop our own databases. So we try to develop our databases. So in case of PGNP, we have exclusively made PP-omics databases or multi-omics databases for genetics and molecular breeding application in PGNP, which has all the information starting from the genomics to metabolomics, means genomics, transcriptomics, proteomics, and metabolomics in this particular database. Similarly, we have made another databases in-house, that is LDDRG, that is legume disease resistant databases, in which you can find all the information related to the disease present in the legumes. So we have targeted here total 10 legume plants, and what are the all sorts of diseases related to these 10 legume plants you can identify through the LDDRG. So this is also in which we are this TBT, THIST has recently launched Indian Biological Database. So it is similar to the NCBI. Earlier we have the National Biotechnology Databases. Now we have IBDC. So now it is mandatory to submit any of the sequencing we are doing, or any protein identification we have done. So all we need to submit to the IDBC for this thing. Also, we have developed some of the tools for the genome assembly. So this is basically we develop for the hybrid genome assembly in our lab, that is nanopore plus Illumina sequencing. And this is based on the haplotype purge of the analysis. So we are also towards the development of tool aspect, because in case of plants we are limited number of softwares available, so we have to develop through our analysis. So in the towards the end of that, I just wanted to conclude with that the multi-omics analysis and gene editing to design a better plants, because earlier the concept was on the one gene thing we are targeting, but now we are looking on the whole genome aspect. We are looking on the broader picture including the whole genome thing, proteome, transcriptome, metabolome, inom, and this is the multi-omics era in which we can collect this information, and then we can plant it that particular seed material according to the field and soil condition, because that also we can capture the information, and finally to the plate of the consumer and the farmers which they wanted to, so that so that the farmers will also get benefit through the high income and also the consumer preference. Yeah, we are designing the crop. So that is the major objective, and utilizing this information we can able to have a disease resistant and nutritional rich various kinds of crop and vegetables and horticulture crops as well. So in case of rice and wheat, you can have this blight resistance, fragrance rice, high grain number, large grain number rice, which is actually quality which farmers are looking on. And similarly, in case of wheat, we have a powdery mildew resistance, high amylopectin content wheat variety. In case of tomato, there is a purple tomato, which is anthocyanin enriched tomato, mildew resistance tomato. In case of soybean, we have high oilic, linoleic content, and citrus, the most important ingredient. So all these things we can mine through this multi-omics approaches, and these are the some examples of crop which is improved by the genome editing techniques. So the takeaway is now this is the era of high throughput phenotyping along with the multi-omics things, including genomics, transcriptomics, proteomics, metabolomics, and ionomics. In genomics, as I and my first slide mentioned that we can perform GWAS, PWAS, GPWAS, marker-assisted selection, QTL, mapping, fine mapping, and bulk segregant analysis. Transcriptomics, we are looking for the single cell as well as the RNA-seq microarray. Uh, and TWAS proteomics, we are doing the peptide sequencing and structure and function variants, PPI, protein-protein interaction, and further pathway analysis we can include. And we can do this together as well through the proteogenomics approaches, and deploying all this data into machine learning and train our models accordingly, applying different statistical approaches, and that leads to the prediction of selection of that desirable trait. So this is the complete strategy of multi-omics data integration between different layers of omics data to dissect the complex agronomic traits and phenotype prediction by using AI ML approaches. So this is the message from my side. So I would like to thank my people, my students, those who involved, my collaborator and my funding agency for this work, support me in different projects. And with this, I would like to finish my talk. So thank you so much for your patient listening. And if you have any questions, I would be happy to take.
So, thank you so much, Dr. Nisha. It was a very knowledgeable session on multi-omics technology in enhancing crop yields while also having a significant nutritional value. So, it's time to take some questions now. If you have any questions, please post them. So, the first question is
Yeah,
the 50
somebody is. Yeah, is the 50K chip is commercially available? Yes, it is commercially available. It is already in the Thermos catalog. You have to order for the rice thing, and you can. It's with the name of OS SMT NKS, so you can order that chip directly. And at a time, you can genotype 96 samples.
Okay. Can we have the next question, please? Hello, could you please elaborate on GPEWAS?
Okay, so GPEWAS is actually your genotyping data, then phenotyping data. Earlier, we are doing with that only GLM model. There are different models are available. So now in the GPEWAS is actually we are considering the environmental factor as well. So the model is changing. So you can either make your own model, or in the GWAS model you have different sets of model like fast GWAS. You have already super GWAS model, farm CPU method. So according to your crop or the organism which you are studying and the ploidy, you can select the model, and that is not one thing you have to do. You have to run on a different models and see how it is doing. If the results is consistent, if your SNP Yes, consistently present in the different models or may vary from the other models, so that you need to check. So there is no, I cannot say that the MLM working well in rice, it will work really well in wheat, because for wheat we are using different model, because wheat is starting from diploid to hexaploid also. So that again depend upon the crop and the material you're using for the analysis. But yes, this is the addition of environmental factor which we earlier ignoring.
Thank you. The next question is, could you please elaborate on the statistical models used in GWAS?
Okay, so statistical model, so different statistical models are there. As I mentioned previously also, initially we have in GWAS when we were doing with the TESOL only almost 10 years ago, that time only two models were there, GLM, generalized linear model, and mixed linear model. But now, because of this environmental factor, and also the other tools like RMVP is there, you have TESOL is there, you have Gapit software is there for the Jiva. So number of tools has already been evolved. So they have different models for that. So accordingly, you have to choose. Also, the major consideration with the Jivas is that you have to take independent data of 3 years. Sometime people what they do, they have sum up the 3 years data and then perform the Jivas, which is a wrong approach. You have to perform the independent the association analysis and see that within these 3 years how consistent your SNPs is present in the association, and that SNP is actually a true SNP, and rest is the false discovery. So that's how you have to filter your data.
Thank you. Could we have the next question is, what tools are used for development of high density linkage map and how can they be interpreted?
Yeah. So for the high density linkage map, there is a tool which is paid. It's a join map. Also, there is another tool, ICI mapping. So here in house we have join map tool for the linkage analysis.
Thank you.
And how you can interpret? So for that you have to have a like your lines P1, P2, and based on that you have data. So you can generate. It's a linkage mapping. So you can generate the linkage based on the recombination frequency.
Thank you. Is there another question? Is syntenic analysis the same as phylogenetic?
No, no. Syntenic analysis and phylogenetic analysis is actually two different analysis. In syntenic analysis, we generally do the chromosome relocation arrangement. In phylogenetic analysis, we study across the species, which I have shown. And synteny analysis generally between at a time one or two species, but in phylogenetic analysis you can have a multiple species of whole genome sequence. Also you can trace the evolutionary time, so you can do the molecular dating and clocking, which is not present in the synteny analysis.
Okay, thank you. Um, which is the best high throughput phenotyping platform?
Okay, so high throughput phenotyping platform again based on the like what trait you want to map. Like for example, if you want to capture the leaf size, if you want to know the like plant height, so that way you have to choose ki you want to want a drone, or you want to have a robots running in your field, or maybe some optical IoT based thing you want to do through which you can perform the phenotyping. So which trait you are targeting, it's all depend upon that. You have to choose the suitable platform.
Okay. So I think we are done with our questions. So yeah, thank you so much, ma'am.
Thank you.
So it's now time to felicitate our speaker of the day with a certificate of appreciation. So thank you so much, ma'am, once again for giving your time and for the great interaction. It was a pleasure having you with us here today.
Thank you. Thank you so much.
So I'd also like to thank the participants as well. So if you guys want more such sessions, please do subscribe to our channel and do leave a like. So yeah, thank you so much everyone once again, and thank you so much, ma'am.
Okay, so yeah, so thank you so much, the Altum team and Sheetal and Simran and Dr. Divya. All of you invited me and sharing my science here. So thank you so much for this opportunity, and I really wish you all happy International Women's Day from my side. So thank you.
Thank you, ma'am.